look at phase 2 studies and you select a dose based upon one of the trials, and then let's say you move into a phase 3 study and you find that that study shows results that are different, substantially different from the phase 2 study. Do you see that those are related to study design? And if there are, do you have any guidance for the people that are here today in terms of what parameters they ought to consider varying or reconsidering in terms of recalibrating the study for what will likely be a repeat?
The typical thing that we see different between phase 2 and phase 3 are straight numbers. And so to the extent that you have larger numbers, you probably have a more accurate assessment in your trial that has larger numbers. So to that extent, yes, I think it's useful to then learn from the trial that you have the larger numbers because it's more likely to be representative. Is it always true? No. But I typically advise looking for the trials where you've seen the most patient responses and use that information to guide your next trial.
Let me just continue one additional add-on there, if I may. Then, is there a possibility for a phase 2-3 design in dry eye trials if the phase 2 studies are not going to be predictive of what the concentration that may be effective is going to be? Do you see an opportunity for combining the two phases and making a single trial that would be large enough to be valuable?
Yeah, as I had listed in my talk, the requirement is to show inadequate and well-controlled trials that a product is safe and efficacious. It doesn't say to show inadequate and well-controlled phase 3 trials. You can do a trial that you think is a phase 1 trial, and if it's an adequate and well-controlled trial and it shows safety and efficacy, and then you repeat that, that can count toward supporting approval. It's a matter of gamble. And do you want to risk at phase 2 that you know what the right concentration is and are going to expose enough patients right off the bat to be able to show safety and efficacy? If you think you have priors, if you think you have animal work to support going directly to a phase 2, phase 3 trial, most of the time we won't stop that.
Mark, I think it sort of flows from what this discussion is, and that is the issue that many of you may be aware of, and that is the terminology adaptive clinical trial. How we use that, how we apply it, and how, Dr. Chambers, you see this from the side of the drug development process that, of course, Jerry and Les have been involved with for combined 65 years or something like that. You have animal data, you have a target, you have a drug, you have stability, and now you're looking to see what subgroup. Is it a mild, a moderate, or severe? Is it a certain combination of diseases that this drug will work best in? As well, of course, as dosing and frequency. But what endpoints are going to be most affected, the early indicators, and what will be the time points as you test them at, are all a multitude of questions that I guess Katie would say there's about 100 variables in there, and how do you narrow that down before you embark on a multi-million-dollar year or longer study and assure the fact that at least you have narrowed the target point? And so in many ways, what you do early towards getting to proof of concept is what this is all about. Once you get to proof of concept, I think the pathway appears relatively clear. But let's discuss for a moment, because that's of interest to all of us here. What do we feel, and I would address this to Les and Jerry and George, would be the best approach and adaptive process to get to POC, time and cost effective, with the highest likelihood of success to either say your new chemical entity works or not? And number two, Dr. Chambers, what where are the sidelines in terms of what can be accomplished under the rubric of adaptive clinical trials?